All right, I think we'll go ahead and get started. Welcome everyone to the Allen Institute. We're so glad you could join us to help celebrate our 20th anniversary. My name is Claire Weichselbaum and I'm a member of the Education and Engagement team here. And we are very excited to share some of our open science with you all tonight. I want to just briefly let you know kind of the plan for the evening. So we're going to have a series of short talks from one scientist in each of our divisions. And then we will have a few minutes for Q&A. So you should all have received a note card and a pencil when you came in. Feel free to jot down any questions throughout the talks. After each talk, we will have folks walking up and down the aisles with baskets to collect those. Just pass it on down. If you need a fresh note card, if you have more questions, just raise your hand and they will hand you one. And then after our Q&A, we will head into the atrium for those of us who are here in person. For some refreshments and an opportunity to kind of get hands-on and really explore some of our open data and tools up close. So without further ado, I would like to turn it over to our CEO and president, Dr. Rui Costa, for some opening remarks. Thank you.
Thank you. And thank you everyone for being here. We're celebrating 20 years of impact at the Allen Institute. Our anniversary was actually September 16th during Open Science Week, but more and more we want to make every week open science at the Allen. And by open science, we mean many things, and we'll share a little bit what we mean here. But we also mean sharing our findings with society in general and trying to explain the things we do in an accessible manner. And what is it that we do? So the Allen Institute was founded 20 years ago by Paul Allen, and now supported by Jody Allen, and we're going to be about 800 by the end of the year, and what we do is we go after the biggest questions in life. And in biology and in health, and usually questions that maybe smaller groups of researchers cannot tackle, or things that are difficult and need interdisciplinary teams. And then we actually openly share not only what we find, but all our data and tools and standards, right? So we call these the Allen Way, and let's dive a little bit about the Allen Way, right? So big science, what does it mean? Think of like moonshot projects or Manhattan-style projects, right? So when we go to study the brain, we decided, hey, do we know all the parts of the brain, all the cell types? Let's start with this, a list of all the parts of all the cell types. Can we do that? And now we have a parts list. In the immune system, we thought, what's the normal immune system of a teenager, a young adult, an older adult, and what happens when there is in disease? Could we profile, with great detail what happens inside our immune system? The same with individual cells. Individual cells have tons of little components and organelles and proteins. What's normal? So these projects are sometimes too big to be tackled by a scientist or even a small lab. So because they are big, we do it with teams of the interdisciplinary researchers, engineers, biologists, people in optics, computational scientists, all working together. Team science also means we have each other's back, and we can go further, hopefully. and open science. And open science, which we're celebrating here today, and it means we share. We share what we find, but we share the data so others can find, which is a little bit important. So we share the discovery progress. And finally, we want to have impact. An impact, of course, can be in health, curing and preventing things. I mean, we know big afflictions beyond just climate change are, our mental health, for example. By 2050, 1 in every 3 of us may have a brain problem. We're aging in a good way. Our life expectancy has increased, but cell senescence and our immune system, as we're finding out, need to cope with all these. So we want to have impact and be responsive to problems in society. But understanding how things work can also lead us to have, understanding how the brain works, to have better educational programs. A lot of the findings we had on cholesterol that give us now a pill, but a lot of life changes have happened based on basic science of what you're supposed to do to prevent heart disease, for example. Anyway, so these are our core principles, and as you probably suspect by now, we have 4 institutes here, right? One is the Allen Institute for Brain Science, which is characterizing these parts list of the brain. The Allen Institute for Cell Science, looking at those details in the cells. For immunology, for neural dynamics, looking how all those parts, all those cells and circuits work together to produce even a simple decision or a simple movement of the arm. And then we have the Paul Allen's Frontier Group, which is a group that actually funds researchers outside of our building to actually also tackle transformative questions. And they are all throughout the nation and all throughout the world. And here are some key milestones, and I just want to point to you. For example, the first institute to be launched was the Allen Institute for Brain Science, and very quickly you see a release of a mouse brain atlas, human brain atlas, connectivity atlas. We launched the Allen Institute for Cell Science, and you see Allen Cell Explorer being openly sharing things with the world. We share an Alzheimer brain cell atlas. We launched the Allen Institute for Immunology, and we shared the data and tools, the Human Immune System Explorer. And with the Paul Allen Frontiers Group, for example, one of the investigators we funded got a Nobel Prize for Human Evolution last year. So these are just some of the things of the last 20 years. There's a lot to unpack, but what you see is the launching of a big program, and some years later, a sharing with the world, a massive data set, and also the tools for people to actually do further discoveries. In some data sets, there are more papers from outside researchers, more publications, more findings in the world from outside researchers than ourselves. And we think that's good, right? We, imagine if we went to the moon and we observed things and then came back and like, what did you see? It was like, oh, we've seen it, we'll share when we're ready. No, we're sharing. In the Alzheimer's disease project, we're sharing even sometimes before there is a publication because there's such urgency for people to have this data. Anyway, so as I said, we developed, and share tools, methods, models, data standards, our findings in translational research. And our next steps are actually to share complete ways, complete platforms in which people can come in and explore the data and do discoveries themselves. This is all I had to say. Of course, we don't do this alone. More and more, we work as a part of a large network of partners. We are a hub. We're very proud to be here in Seattle and South Lake Union. These are some of our partners that you may recognize. And one of the big partners, we hope, is the local community. So anyway, thank you, and let's get into the real program.
Well, good evening, everyone. I'm Megan Quinlan, and I'm a scientist on the gene therapy team. And it's truly an honor to be the one kicking off this showcase of all the different science units here at the Allen Institute. And we'll start with brain science. Tonight, I want to highlight two things for you. One is what the Allen Institute for Brain Science is probably most known for, and that's these detailed maps of the mouse, human, and primate brain. And two, how my team is now using these maps to potentially treat complex brain disorders. But you might be wondering, what do we mean by quote unquote mapping the brain, and why are these maps so important? Well, I think we can all agree our brains are incredibly intricate structures that house billions of brain cells called neurons. And all these star-like shaped cells are responsible for our thoughts, emotions, and behaviors. I know for me personally, understanding how these cells work together to create the human experience is what drew me to studying the brain. And one tool to help answer all these complex questions about the brain is a comprehensive, detailed map of it. Just like you would use a map to help you navigate a city. We can think of the map from many different levels though, right? For example, we can divide our map up into different neighborhoods, like Ballard versus South Lake Union, and within the brain, we can also divide it up into many different brain regions, like you might have heard of the hippocampus, cerebellum, and cortex. And while these types of maps have been available to neuroscientists for a long time, and they're very useful, we can still get even more detailed, right? Just like this map shows us where different buildings are located, we can do the same to show where different types of brain cells are located within the brain. Now, some of you may have heard there are different types of brain cells, like neurons and glia. So for our map analogy, that would be like different types of buildings on our map, where neurons are stores and glia, we could say, are a hospital. But once again, we can get a little bit more granular. Just like we may classify different types of stores on our map, like a retail store versus a grocery store, we also have many different types of neurons within our brain. But how do we categorize all these different types of neurons? And that's where our journey begins. The Allen Institute for Brain Science embarked on a mission to map these diverse cell types within the brain. To do this, though, we first need to understand the language of our cells, which is written in our DNA. Specifically, the part of our DNA called genes. So you may have heard that genes influence who we are, and that's absolutely true. While every cell in our body contains the same DNA, what sets each cell apart is which genes in that cell are read to make proteins. Think of it like a store ordering products to sell. Every store can order the same thing, but what they choose to put on their shelf is what really sets them apart. So for example, these two neurons that express very different genes, like the one here in light blue versus the orange neuron, are less similar and can be compared to like a home improvement store versus a clothing store. While cells that are more similar might be like comparing a Trader Joe's to a Safeway or Whole Foods. There are subtle differences, but these differences are actually crucial in determining where you'll go shop. And it's the same in the brain. Different neurons have different sets of genes turned on, giving them their unique characteristics, functions, and locations within the brain. We have now cataloged over 5,200 different types of cells in the brain, and I don't think I even fully appreciated the cellular diversity of the brain until I joined the Allen Institute. And thanks to a monumental effort, the brain sciences have now launched their newest map, the Allen Brain Cell Atlas, or ABC, showcasing this brain's cellular diversity and where all these different cell types are spatially located in the brain. This video shows you a cross-section of a mouse brain, and each color is a different cell type. And as we zoom out, you can see all these cross-sections across the entire mouse brain, giving us a detailed understanding of where in the brain which genes are expressed in what cell types. You can even explore this for yourself at brainmap.org. By the way, I encourage you all to take a look at these maps yourself during the events tonight or online when you go home. You can explore numerous different brain atlases that brainmap.org has to offer. It's a freely available resource that neuroscientists worldwide rely on. These brain maps is how I knew about the Allen Institute before I joined, as I used them myself in other labs to know where specific genes were expressed in the brain. But our journey doesn't stop with just mapping the brain cellular diversity. There are numerous talented teams within brain science who are continually expanding on these brain atlases across different animals, ages, and diseases. But today, I want to shine a spotlight on my team, Gene Therapy, where we are now leveraging these maps to develop therapies for brain disorders. Oops. Our primary focus within the gene therapy team is on disorders with known genetic causes, where a specific gene is mutated, leading to a reduced expression or loss of its gene's proper function. Our team's mission is to investigate whether providing a functional copy of that missing or mutated gene can effectively cure the disease by addressing the root cause of it. But we first need to know where that gene is expressed, and that's where our maps become invaluable, right? Since we know which cell types a gene is expressed in, we can now deliver our gene only to the cells where they are needed, effectively providing an address for that gene. This way, the gene is only expressed where it should be. This cell type specific gene therapy approach allows us to treat disorders with greater precision and fewer side effects. To show you the real power of this strategy, I want to tell you about a targeted gene therapy we are developing for Dravet syndrome. While you may have never heard of Dravet syndrome because it's a very rare genetic disease, only affecting about 1 in 16,000 patients in the US, it does lead to treatment-resistant epilepsy in very young children, with most patients' seizures starting before their first birthday. Unfortunately, about 10 to 20% of children do not survive into adulthood with Dravet syndrome. So it's a very devastating condition, and it's caused by the loss of one critical gene, SCN1A. Our team, in collaboration with Seattle Children's, took on this challenge to see if giving back the SCN1A gene with the cell type specific tag, meaning it will only be expressed in the cells where it should be, can we treat some of the symptoms of this disease? But before we can test this in humans, we need a preclinical model, which for us is mice that have also lost their SCN1 gene. And as you can see on the graph on the right, the mice without the SCN1A gene, more than half do not live past 70 days. So you may be thinking that's not a very long time, but mice don't live as long as humans. By 6 weeks of age, mice are already considered adults. But remarkably, though, the mice that did receive our cell type specific SCN1A gene, every single one of them survived. Importantly, when we provided this gene to all the neurons, we did not see this same level of success. We also have the ability to measure seizures in these mice, and the mice that received the cell type specific SCN1A gene therapy showed a significant reduction in the number of seizures, a truly promising result. Now, while this progress has only been achieved in mice, it does hold immense promise for human patients. This strategy may not only be effective for Dravet syndrome, but also for other brain disorders that have genetic root causes. This offers hope to countless individuals and families affected by these conditions. In conclusion, I hope today that I showed you that in order for us in labs all over the world to be able to study the brain in all its complexity, a helpful tool is a detailed map of its cellular diversity. And two, now that we have these intricate brain maps in place, we are opening the doors for effective, targeted, and personalized treatment for brain disorders, a remarkable feat that is undoubtedly shaping the future of neuroscience and medicine. And with that, I want to first thank you all for joining me today. I also want to give a big shout out to the gene therapy team, specifically Drs. Ed Lean, Boaz Levy, John Mitch, Brian Gore, and Ron Go, who are here at the Allen Institute for Brain Science, and our collaborators, Franck Coloumé at Seattle Children's, who are all instrumental in that Dravet story. And last but certainly not least, I want to extend my gratitude to the whole team at the Allen Institute for Brain Science, especially Dr. Honghui Zhang, and the generous funding through the NIH Brain Initiative. This work would have not been possible without the dedication of many talented individuals collaborating together. It has unlocked the concept of big science, making it accessible to the public, and is ushering us into this new exciting era of potentially treating brain disorders. Thank you.
Hello, everyone. and thanks for being here or in front of your computers. My name is Gökhan Dalgın. I'm a senior scientist at the Cell Science Institute, and I would like to shift the focus from how different brain cells and intricate networking of these cells actually generates a functional organ to how deep and holistic understanding of the building blocks of life, the cell, is critical for understanding and solving the mysteries of biology and also helping to advance human health. And to do that, at the Institute, we use human-induced pluripotent stem cells. So why do we do this? Our goal is to understand the organization and behavior of cells. There is a gap in this knowledge as researchers often focus on a few components of the cell in one context and affecting holistic and dynamic understanding of human cells. So now let's look at how we set out to achieve this ambitious goal. Some of you may have already seen how a cell looks like in a textbook, and this beautiful depiction that you see as a diagram. And you can see that this diagram shows nice organization of the different structures in the cells. The one that you see in the center is a blue nucleus, and then other structures around it. So in contrast to this diagram, human-induced pluripotent stem cells, iPSCs in short, these are the cell types that we use at the Institute, as I told you, look a lot different under a scope. iPSCs do not look colorful. We make them colorful to see under the scope, but I will tell you more about that, how we do this later. You can see a live cell imaging of a single cell, and it is dynamic behavior, such as in green is the nucleus, and the rest of the cell is in magenta. You can already appreciate that this cell looks a lot different than the diagram on the left, and you see dynamic changes of nucleus. In fact, we imaged and analyzed over 200,000 iPSCs to build a new mathematical framework to understand our cellular building blocks. As you can see in this picture, that iPSCs can have many different shapes and different organizations. We not only take these beautiful images, we also use these images to create computational results and models like the one you see in here, showing first time ever an intracellular organization of an iPSC. This research is published in Nature, and the data are open on our website and to everyone who wants to use it. Now let me tell you how we generate these beautiful cells and images, beginning with how iPSCs are made. So these iPSCs are generated by skin samples, and it can be taken from anyone, including yourself. But we use a program called reprogramming that can take the skin cells, which only function as skin cells, but this reprogramming turned these skin cells to pluripotent stem cells. Now these cells have the potential to become any type of cells. So we house these normal iPSCs at the Institute, but then we use another tool called CRISPR-Cas9 gene editing, and you might actually heard that in the media or social media. And we actually use a DNA sequence which comes from jellyfish, actually from Puget Sound, and then insert this by using this gene editing into a desired location to eliminate different structures of the nucleus. And then we use elaborate imaging techniques, different strong, powerful microscopes to get images like this. The one you see in here is illuminating the nucleus. And, oops. When I play this, you can see that nucleus are in different shape. And then also you can see that one cell is becoming a two cell. And this actually shows that how cells divide. It's called cell division. And at the reception, my colleague Gideon here will actually help you to understand how different steps are going through from becoming a one cell to two cells by showing you a nice game that you can actually put a VR sets and then play with it. It's called mitosis. So I would like to also mention that every step in this process has previously won a Nobel Prize, not by us, by other researchers. But I would like to stress in here that how basic science is actually instrumental to help other researchers to discover new biology and ultimately affecting and helping to improve human health. And if this actually, the linear progression of winning Nobel Prize is correct, then we might be up for winning another Nobel Prize in the near future. Pressure is on. So this process that I showed you so far actually being used, and we generated over 50 lines, which is marking the different structures in the cells. And you can see that these are different and beautiful structures. And it is much different than what you have seen in the first original depiction of the cartoon that I showed you before. So we make these cell lines available for researchers around the world. So far, we have distributed all of these countries shown in purple. In fact, most recently, one of our gene-edited cells has reached into the space to be studied in the International Space Station to understand the effect of microgravity in iPSC's biology. So one might actually think that we might be working with little green cells. Now I will tell you a little bit more on how we actually use these cells and to do research at the Institute. So what we can do is actually we can take a ball of iPSCs and change them to different cell types, which I told you earlier, because these cells have the potential to become different type of cells in your body. And if you look at this, the image on the left is showing you how induced pluripotent stem cells look like. And then when we change them to human endothelial cells, which actually covers the lining of your blood vessels, although these are coming from the same line, you can see these structures are completely different than each other. And we can do this by changing the different cell types. It's called differentiation. But differentiation doesn't happen in a single step. It actually happens in multiple steps. And I would like to tell you two short stories how we use these different steps to understand how cells organize, behave, and change. So in the first part of the differentiation stage, we can actually take this ball of iPSC cells, as I showed you earlier, and normal iPSC cells doesn't live alone, they like to live together, and they are called iPSC colonies. So at the beginning of this process, we can actually sprinkle some cytokines or growth factors to push iPSCs to become pre-endothelial stage. And then in this time-lapse movie, what you're going to see is that this colony behavior will change upon receiving these cytokines. You will see that this colony will contract and then comes to maximum contraction. And after that, some cells will start to move away from the colony, and you will see a massive migration. Display that. Okay, now you're seeing a colony-wise change in the cellular shape, which comes to a massive migratory cell. And you can appreciate that these behaviors are a lot to follow. So there is a lot going on here, and it is possible for scientists to actually focus on a certain time in point of this differentiation than to understand the entire at once. So what we do first, we focus on the migratory cell stage. And in here, you're just seeing a few cells, which the nucleus in green. And we use computer algorithms to define different behaviors. And then we define that behaviors to their behavior and biology. So when I start this movie, you will see that in this colored order, the cells that are not in the colony, they are not moving, and then they will start moving and dissociate from the colony. And then after they dissociate it, they will have an inhibition because they will touch to another cell, and ultimately they will start to explore the debris. Now the cell actually has moved, and the trajectory is going towards and away from the colony. It changed rotation, and then there is the locomotion, and then you can see that it's exploring the debris right now. So we can actually use this and start to understand and build a framework how this intricate behavior of the cells moving away from the colony leads up to generating pre-endothelial cells. I think this is even more than that, just to understand how normal cell functions. It is also important to notice that these type of behaviors not only happen in normal behavior, it also happens in pathological conditions such as cancer. Therefore, building up a framework to understand how these behaviors and the ground truth to understand and show that what is the variation in the normal cell behavior will help other researchers in their line of work, what is normal, what is different. So now I will lastly show you how we use the second part of this differentiation protocol to understand the changes from pre-endothelial stage to the endothelial stage. So when we actually put more different sprinkles onto the cells, when they are endothelial cells, they are going to change and become pre-endothelial cells, they are going to change and become an endothelial cells. And you can see a lot of endothelial cells in here. They are mixed in their organization, and normally your endothelial cells are exposed to blood flow. And we can actually mimic these conditions, this blood flow, in the lab. Therefore, we can actually apply different flow to these cells. And how do we do that? We can use this chamber slide that you see right underneath, and then we can hook this to a machine where we can generate different strengths of flow that your cells, endothelial cells, are exposed in the body. Normally, there is no constant one flow in your body. It's actually every part of the endothelial cell, whether it's in the brain or the kidney or elsewhere, is exposed to different strengths of the flow. So therefore, we mimic these conditions, and we expose the cells to a low flow, cells align in horizontal way, and we expose cells to high flow. Now you can see that they are perpendicularly aligned. So this might be actually interesting for us because with minimal intervention, now we can actually change the behavior of the cells and start to understand whether these could be associated with some of the pathologies, as we know that the change in the blood vessel might actually cause such as like vessel flow, such as arteriosclerosis. So finally, I would like to thank you for listening, and all work that are shown here would not be possible without the generosity of Paul Allen and hard work of our past and current diverse team members. And I'm happy to take questions at the Q&A or in the reception. Thanks.
All right. Well, it's my pleasure to be here. I'm Troy Torgerson. I'm the Director of Experimental Immunology at the Allen Institute for Immunology. And I want to tell you about work we're doing to work toward trying to understand basic mechanisms of immune diseases by really understanding the mechanisms of how the immune system functions in order that we might ultimately be able to alter the course of chronic diseases. And I'm going to give you an example of one disease that we're working on, which is rheumatoid arthritis. So what we're doing at the Institute is really looking at the immune system in a variety of different ways, from lots of different directions, using cutting-edge technologies that allow us to look at the human immune system in ways that it hasn't been looked at before. And today I'm going to tell you a little bit about the immune system and how the immune system responds to things. I'm going to give you a brief introduction to rheumatoid arthritis, and I'm going to tell you a little bit about one of our interesting recent findings related to rheumatoid arthritis. So, the immune system. So for this, I would like you to imagine that your body is an island, that you are in an island and you're being under constant attack by bacteria and viruses that are coming from every direction. They're coming from the air, from space, to attack your island. And so you need defenses to protect you against all of these invaders. And so you need ground defenses, you need air defenses, you need space defenses, and you need peacekeepers on your island to make it so that there's no infighting among those on the island, right? And so your immune system provides all of these defenses to your body and prevents you from being invaded by all of these pathogens that want to come and take over. Now, what happens under a normal circumstance where the immune system is triggered by an event like an infection is, I think of it like a traffic light. So similar to a traffic light, you pull up to you're at an intersection, the green light goes on, you hit the accelerator, and like that, your immune system following a trigger will accelerate and become activated to respond to, say, a pathogen. And as it becomes activated, it's responding effectively to that pathogen. You're driving down the road, you're coming to the next intersection, and you see the yellow light. And it says, hey, start slowing down. You've accelerated enough. You need to slow down in order to prevent a crash. And so similarly in the immune system, there's a regulatory phase that kicks in and tries to prevent the immune system from overreacting to things. And we heard a lot about this during COVID with cytokine storm and the things that could happen as the immune system became overactivated, okay? So the immune system is slowing down, and then once you've had an effective immune response and you've cleared the pathogen, you need to resolve that immune response so that all of that activity goes back to baseline and returns here to sort of a baseline state. Now, there are immune cells, proteins, lipids, other things that are involved in this immune activation and this regulation and response. And then it ultimately leaves you with a memory response. Your immune system remembers what it has seen, like the brain remembers things that it has seen. The immune system also learns and has memory and remembers those things that it's seen before. Now, this can go wrong in a few different ways, as you can imagine. So you might have overactivation, as represented by the thick green line here, where the immune system just over-responds to an original trigger. You might have under-regulation, so that yellow light that says, start slowing down, just doesn't work effectively, and so the immune system doesn't start slowing down, it just over-responds. Or the resolution mechanisms may not work and may not resolve the immune response that's happened, and that leaves you then with an immune response that persists, and it may lead to autoimmunity or autoinflammation, okay? Now, one of the things that I'd like you to just think about is this, that we ask the immune system to really perform what I think is really an impossible task. And it's amazing that we don't all have problems. And that impossible task is that it's been tasked with really defending us against any possible pathogen, anything that we could possibly see. But we also tell it, you need to do that, but you can't respond against us, right? You can't cause autoimmunity. That seems like an impossible thing that we're asking it to do. Okay, and so I'm going to tell you a little bit about that with regard to this particular immune disease. All right, rheumatoid arthritis. So rheumatoid arthritis affects 18 million people worldwide. It was an estimate in 2019. Women are affected about 3 times more commonly than men, which is common for many autoimmune diseases. About 1/3 of individuals with rheumatoid arthritis have to stop working within 5 years of getting the disease because of joint destruction and disability. And the annual cost in the US economy is about $40 billion to take care of patients with rheumatoid arthritis, okay? Within your lifetime, you've got about a 1% chance of developing rheumatoid arthritis. So what is rheumatoid arthritis? Well, here's a normal joint. So arthritis, of course, is inflammation and warmth around joints and pain. And the joint, here's the bone. The joint has cartilage here that acts as sort of a surface that's smooth. There's There's a synovial membrane, which is sort of very feathery and secretes synovial fluid that's in here that's almost like oil that keeps your joints smoothly moving back and forth. And in the setting of rheumatoid arthritis, for some reason your immune system becomes confused and it attacks the joint. Immune cells move into the joint, they cause overproduction of synovial fluid, the synovial membrane becomes overgrown, infiltration of immune cells, and ultimately it leads to degradation of bone and leads to joint destruction and disability, okay? So we know that patients with rheumatoid arthritis develop autoantibodies, okay? And so what is an antibody? An antibody is a protein that your immune system makes. It makes it in response to pathogens. It makes it in response to vaccines. And this is an antibody. It literally looks like this. It looks like a Y. I think of these as sort of the hot dog roasters of the immune system. So, you know, here's the end that the hot dog goes on. This is where the antibody binds to, say, a pathogen here. Here's where the immune system grabs hold of that and uses that to get rid of that. Now you can have these antibodies that, again, they're not made towards pathogens, they're made toward our own cells, and that's an autoantibody. They shouldn't do that, right? We shouldn't have autoantibodies. Ideally, that's what happens. We know that patients with rheumatoid arthritis can develop a couple kinds of autoantibodies, these anti-citrulline protein antibodies that I'll call ACPAs, and then rheumatoid factor antibodies. I'll show you a little bit of data from one of our collaborators that we're working closely with on this project. This was an interesting study where they took patients who developed rheumatoid arthritis, here in blue. They had controls who didn't, and they asked them, have you ever been in the military? And if they said yes, they went back to the military and they said, we want to look how far back did these patients develop rheumatoid arthritis before they actually developed the active disease, the active arthritis. They wanted to see when the autoantibodies presented. And so they went back to military biobanks from basic training, and they collected samples, and they looked, and it turns out that many of these patients had developed these autoantibodies 17 years, as long as 17 years before they developed active disease. So they had already developed the autoantibodies, but they hadn't developed any active clinical arthritis. And so this period is called the at-risk period for rheumatoid arthritis, and it's identified by the presence of these autoantibodies. And this is one of the projects that we're working on, is focusing on this group of patients, thinking that if we can understand what's going on in this period and how patients convert to active clinical disease, we could understand more about the basic mechanisms of this disease. All right, so let me show you this little piece of data from this study. This is recent data that was a surprise to us, really, because, you know, these patients are well, they're fine, they don't have joint pains, they don't have arthritis, but lo and behold, if you look at, for instance, in their peripheral blood, these are all inflammatory proteins that are being excessively made by these patients. Their immune cells are overactivated, and it looks like they actually have active rheumatoid arthritis, but they don't have active rheumatoid arthritis. So even though they look fine, the inflammation is already there. And using this, we're really trying to mine and understand the mechanisms of disease. The way that I think about this is that it would be as if you had a nice car. It looks great on the outside, but then you open the hood and you go, what the heck is going on? It's like broken inside, right? Things are already like on fire, but the house hasn't burned down yet. And we know that this car is headed for trouble, right? It's going to break down at some point if these antibodies are present. But now we know that really it looks like they've already got a lot of inflammation. So why is this important? Well, if you don't treat patients' rheumatoid arthritis, this was joint damage and disability, they have a lot of problems over time. Original treatment focused on really late stages of disease, and you can sort of stabilize disease, you can treat earlier and stabilize disease. Now that we know that patients have active inflammation early before they develop the actual arthritis, the question is, could we sort of identify these patients and treat this phase of disease and actually prevent onset or delay onset of disease, thus hopefully preventing some of the effects that we talked about earlier. So what have we talked about? So the immune system protects you from all sorts of invaders. It gets activated and deactivated in a very coordinated way, and it shouldn't attack you or your tissues, okay? That's the first thing. That in rheumatoid arthritis, many patients develop autoimmunity long before the presence of active clinical disease, and we're hoping that this period where there's a predictive biomarker may help us to understand how the disease onset takes place, what the causes of that are, and also might be a period during which we might treat patients to prevent or delay the onset of disease, okay? Thank you very much. I want to just acknowledge all of the members of our team who've worked incredibly hard to bring this work forward, and for, of course, our very generous founder who made it possible for us to be able to do this work. So I'll be happy to take any questions. There's a booth outside, and Vaish and Tiana will challenge you to one of the tasks that we do, which is cell sorting, and they'll challenge you to do some cell sorting with them and tell you a little bit about that. So thank you very much.
Okay, let's see. Thanks everybody for sticking to the end. My name is Cindy Poo. I'm a senior scientist here at the Allen Institute for Neurodynamics. So really excited to tell you a little bit about our ongoing work and what we have planned for the future. So the first talk you heard from Megan about maps of cells in the brain. I will now tell you a little bit about our efforts of trying to understand how all these different types of cells dynamically interact with each other in order to generate thoughts and produce behavior. In other words, what we're trying to do is to identify the function of all these different cell types in a living animal. And as you can imagine, this is an incredibly difficult and challenging task. So how do we go about this? Some of you, or many of you, may know that neurons communicate via electricity. So here in this video, what I'm showing you is these flashes. These pink flashes are all triggered by electrical impulses in individual neurons, and they correspond to one neuron communicating with another. What we would like to do is we would like to study these types of network activity as the animal is behaving, perceiving the world, generating thoughts and actions. And to do this, we study mice because they share a lot of the functional organization with humans. Okay, so this is, of course, the desire to measure and understand the brain by measuring its activity is not a new thing. Scientists have been measuring neuroactivity for a long time. So some of you may recognize experiments like this. So in classical neurophysiology experiments, recordings were often done with tungsten electrodes. So these are single wire electrodes with one site on them. If you place them close to a neuron, you can observe the electrical impulses or spikes of of one cell. As you can see here on the left, what we have here is voltage change across time. And these vertical ticks, individual vertical ticks, reflect individual spiking activity or electrical impulses of one cell. Now, of course, as neurotechnology and engineering has advanced over the past decades, our ability to perform these recordings have also scaled up. And they've scaled up not just in terms of the numbers of recording sites that are on each one of these electrodes, but they have also decreased in size. And so they allow us to, in a minimally invasive way, record from hundreds of neurons in the brain simultaneously at a single spike resolution. So this has really transformed the field of systems neuroscience, or the field in which trying to understand the dynamics of how neurons talk to each other. And yeah, so this is what one of these NeuroPixels probes look like. You can see that they're extraordinarily thin, about the thickness similar to a single strand of human hair. And we have one of these probes out in the atrium later on for those of you who are curious to check it out. Okay, so scientists here at the Allen Institute have been using these probes to measure activity from hundreds of neurons in the brain to ask a very fundamental question, which is, how do images that hit our retina, how are these images transformed in the brain to generate visual perception? And they've been doing this by systematically recording from interconnected visual pathways in the mouse brain. And they place these electrodes. So now that we're recording, not just using 1 neuropixels probe, but up to 6 neuropixels probe. And we target them and we place them in the brain in a very careful manner that's guided by the knowledge base and the maps that's generated from our sister institute, the Allen Institute for Brain Science. So this allows us to perform these very targeted recordings from large groups of neurons while mice are viewing a series of carefully designed visual scenes or movies with distinct statistical properties. And there are a few foundational discoveries that are made from these experiments. So first of all, what we found was that functionality followed the anatomy. And what I mean by that is neurons in areas of the brain that were the closest to the retina were earlier on in the visual pathway responded the earliest when we flashed an impulse of light in front of the mouse. And you can see that from this pink trace here, which is an area of the brain early on in the visual pathway. And the neurons that are higher up in the hierarchy, in places where we think kind of perceptual properties of vision is generated, responded later. And they also were hypothesized to be later on and higher up in the hierarchy. So functionality followed anatomical hierarchy. But perhaps most excitingly is because we were able to record from all of these neurons simultaneously, we can probe their dynamical interaction. And what we found is that and actually for the first time, this is the first set of experiments in which we were able to simultaneously measure activity throughout the visual pathway. And what we were able to characterize and understand the flow of forward and backward flow of information throughout this pathway dynamically as animals were watching these different visual scenes. So in this data set, 100 neurons were recorded. Now, this is a massive data set and a wealth of knowledge in here. And this goes back to a core principle that all of our speakers highlighted today, including Rui earlier on, which is that one of our goals is to generate a rich data set that is accessible to the community at large. And so this data set, along with many other similar ones, were released sequentially as they were collected over time. And what I'm showing you here is a plot of the cumulative numbers of scientific articles that are published reusing this data set. So up to this date, more than 100 articles have been published using this data set, using these data sets. And this is extraordinary. So because this means that not only are we doing big science and open science, but it actually has a real impact to the community. And one of all these different publications span different types of work, right? So they go from groups making new discoveries that we did not look into, or they use this data set to form new hypotheses, or perhaps they used it to test models that they were building to test their predictions. One example of this work I'm going to show you here. So this is one of these reuse cases where scientists use this large data set to actually generate a new machine learning and artificial intelligence tool, which allowed scientists to parse the neural data in a way that allowed us to predict which frame in this movie that the mouse was viewing on a millisecond time scale. Okay, so this is happening in real time, and this is happening in real time, moment by moment. Okay, so this highlights the very tangible impact that our open science has on the community. So moving forward, our teams are very excited about applying these approaches to tackle complex brain functions. And what do I mean by that? So, for example, how do we make decisions in an uncertain world as things are dynamically changing and perhaps not entirely observable or predictable? For example, this mouse that's foraging happily in a grassy field not only needs to explore where all the potential food sources are, but also perhaps remember where it had previously encountered a threat. It needs to do this, but also needs to pay attention to any sudden movements or unpredicted events that may occur. So it needs to kind of hold all these different streams of information in its head simultaneously in order to make a decision about how best to forage for resources here. This is a very complex decision, but it's natural and animals distinctively produce these behaviors. So the idea here is that by using these naturalistic behaviors, we can study computations that are important for flexible decision-making, which is the core basis of cognition and intelligence. And to do these types of experiments, you can imagine we are not out in the grassy fields tracking mice and putting cats in holes. But to do this, as a team of engineers and scientists here have designed a virtual reality system for mice. So here we can very carefully control the entire environment of the mouse, and monitor their behavior, as well as record from interconnected hundreds of neurons throughout the brain, and really try to understand what is the neural basis of decisions, thoughts, and actions. Okay, so ultimately, as I mentioned at the very beginning, what we would like to do is to take what we learn by studying the neurodynamics and connect it back to the foundational knowledge that's generated by the Allen Cell Types Atlas, and this will allow us to leverage the molecular genetic tools and various therapeutic tools that we have to make interventions when things go awry. Okay, so thank you very much for your attention. I would like to acknowledge our entire team. I feel incredibly privileged to work in such an inspiring team full of brilliant minds and collaborative team spirit. So thank you all for your attention, and I'm happy to take any questions.
All right, thank you to all our speakers. Before we move into our Q&A, I just wanted to reiterate what Rui said at the beginning and what all of our speakers have echoed, that the Allen Institute's Open Science is a collaborative effort of many people here in Seattle and around the world. And the growing scale of our scientific impact is made possible by the growing scale of our collaborations and everyone who uses our open data and tools. So thank you all for joining us tonight and being part of this growing Allen Institute community. And now I'd like to invite our speakers to come up, and we have time for just a couple questions. We've gotten a few. And up here, and you're gonna. Oh yeah, perfect. Awesome. All right. So let's start with chatting a little more about open science. So how do you think the Allen Institute's open science approach has changed your work? And any of you feel free to start. You don't not to put you on the spot, Megan.
On the spot. Well, yeah, I think I kind of talked about this in my talk a little bit. I knew about the Allen Institute even before I came here because they have all of these resources. I don't know a single scientist in the neuroscience field that hasn't heard of it and have used all of those brain maps to have as their first figure in a paper. So it's definitely been very helpful for me.
You want to go? I think it's in multiple folds. One of them is that I agree with Megan. That even before I came here, I was aware of all of the institutes, but back then, some of the institutes were not here. The tools and products they had, and I personally used those products, and I truly appreciate it because they were freely provided to the entire scientific community. And I was also impressed by how these, for example, for the Cell Science Institute, lines are actually distributed around the world, and people are using the same line to do different experiments, and these actually have the same quality of same lines. And you don't have to go through the intricacies of generating a line and tagging. It's very elaborate, and it is just, in here, it's much more straightforward.
Anyone else? We're ready for another question. Awesome. All right. So several of you touched on clinical applications down the road of the work that you're doing, and I'm curious both what you see as sort of the ultimate future for the work you're doing, if that is in the clinic, but also what do you see as being the value of basic research that maybe doesn't have a direct clinical application?
Okay. So I think we're studying basic mechanisms of how the immune system works in the Allen Institute for Immunology, and we're trying to understand both the basic mechanisms, but that then often leads you to, how could we change or perturb this mechanism to actually modify how the immune system functions, and to really understand how we can prevent or possibly correct disease, as y'all are working on. I think that for us, for the Institute for Immunology, we sort of have both tasks that we had taken on as our deliverables. That there was a basic task that we needed to understand the immune system more, and a task to then to translate that back to people. But for that, that's the Institute for Immunology. That's sort of how at the foundation of what we do.
So yeah, one of the kind of big projects that I didn't have time to tell you about in neurodynamics is setting out to understand all the different neuromodulatory systems in the brain across different timescales. So I think for us, that is a very real and direct way in which we can have an impact on human health, because a lot of the mental health diseases involve a dysregulation or dysfunction in our neuromodulatory system. So neuromodulators are things like dopamine, serotonin, things that you maybe have heard of. But we have a very, very rudimentary understanding of how these molecules function in the brain across various different timescales. And I think this is a big reason why we don't really have really good treatment offers for people who are suffering from mental health disorders. Yeah, so hopefully a basic understanding of what these systems are actually doing individually, and also how they interact with each other. How does dopamine interact with serotonin, for example, will help us make new leaps in terms of therapeutic tools.
Wonderful.
I would just add, very quickly too, that I think Gökhan highlighted really a tool like CRISPR, which was really a basic science discovery. It was that's a bacterial enzyme really, and how basic can you get? That now is being used to edit genes in humans and to treat diseases. And we never know how basic discoveries are ultimately going to be translated to direct applicability. So I think that's an important point to make.
Yeah,
yes,
please.
Not to the CRISPR, but I want to also mention that when one thinks about normal, it's often non-pathogenic. So normal can be in different, like a large variation within the normal, and defining what that normal is and generating a ground truth for that normal will be helpful to scientists, to other scientists where they are, studying pathologies and understand which, for example, in our case, cell will generate a pathology, which one will not. And I think that's a great tool.
Yeah, I guess with mine, it was pretty applicable because we're actually now trying to treat diseases with our cell type map. So it's kind of what we actually saw with the brain maps, we're now translating that, and that's kind of the next goal that we were trying to say. Is we have all this great basic research that the Allen has done, but now how can we move it to the clinic? And that's what we're trying to actually do and show the power of cell type specific gene therapy, right? That hasn't been shown before, and that's what we're hopefully pushing the field towards right now.
Fantastic. The power of basic science that eventually leads to these wonderful applications. All right, well, I think we're out of time, but I encourage all of you to chat with all of our speakers during the reception. Those of you online, thank you so much for joining us as well, and we hope we'll be able to host you here at the Institute sometime soon. I just wanted to quickly mention that we're excited to offer more public events like this in 2024. So you all should receive an email survey to give feedback. please let us know what you enjoyed or not about tonight, and ideas you have for what we might do in the coming year. We're excited for the next 20 years and beyond of the Allen Institute's story. So thank you again for joining us.
Thank you.